RNA, a marvelous tool. Let's start off with DNA. A double helix, the human genome that consists of 4 letters, the bases. It's a huge data set of all hereditary instructions, a source of information for the construction and reconstruction of cells. During mitosis, skin cells require other information than liver or heart muscle cells. DNA is the stored blueprint of life. It is what defines us as human beings. But there is a parallel universe to DNA. RNA, molecules that read and copy specific DNA information in order to produce or encode proteins in accordance with the transcribed information. The human blueprint is a mix of data sets and protein copy jobs. Apart from the protein coding RNAs, there is also a particularly interesting field of non-coding RNAs. Still not fully explored to this day, it makes up the bulk of the genome. Among the types of non-coding RNAs, there are small microRNAs and long non-coding RNAs. All non-coding microRNAs act as guardians, controlling several protein-coding RNAs like conductors in an orchestra, and thus have a great influence on the design of a large number of proteins. At the Institute of Molecular and Translational Therapeutic Strategies, Professor Thum's research team studies non-coding RNAs. They focus on the questions how non-coding RNAs control processes in the heart, and how they can be utilized to cure heart diseases. The scientists examined various scenarios of cardiac strain, oxygen deficiency that can lead to cardiac injury, or cardiac pressure overload that can cause irregular heart growth, referred to as pathological hypertrophy. What are the underlying processes that take place in the heart? The aim is to identify non-coding RNAs that accompany such harmful transformation processes, and that ideally could be implemented in a therapeutic approach. The aim of this research is to produce a non-coding RNA modulator, a drug to treat pathological heart growth, for example, following a heart attack or due to long-term high blood pressure. For instance, microRNA blockers could block the respective docking station within the target cells and thus improve their function. Chemists synthesize these microRNA blockers and use chemical manipulations to ensure that the blockers are taken up by the cells and that aren't metabolized by the body during medical treatment. The therapy is successful if the heart no longer develops the pathological transformation process and resulting functional insufficiency in reaction to the disease. Clinically, beneficial effects can be observed by biomarkers and cardiac imaging techniques such as echocardiography or magnetic resonance imaging. The hope is that these novel, next-generation, non-coding RNA-based drugs will provide better treatment for various heart diseases, which could significantly improve the life expectancy and quality of patients suffering from such detrimental conditions.